Moving on now from our plants, talking about a few other organisms out there now that we've covered animals and plants. Now time to talk about some prokaryotic organisms and some viruses. First of all, our simplest organisms, these are the prokaryotes. Again, prokaryotes do not have a nucleus. They don't have organelles like mitochondria or nucleus, anything like that. So they are termed the simplest, however, the most abundant organisms on the earth. So counting them all up, there are way more prokaryotic than eukaryotic organisms. And they're divided into two groups, bacteria, that's mainly what we're going to focus on, but then also archaea. Give you an idea of the variety of these prokaryotic organisms. Some of them have this rod-shaped, they're called bacillus, we'll see in the next slide. Some of them are spherical or round, called coccus. Some of them can be spiral-shaped. Some are flagellated, meaning they have these long tail-like structure called flagella. Some are actually multi-celled. The majority are single-celled. One cell makes up the whole organism. But these cyanobacteria, they have lots of cells that come together to form the organism, or these stalked bacteria that, again, multiple cells coming together to form the organism. The simplest form, again, the rod shapes are called bacillus, spherical or called cocci or coccus, and then the spiral or coiled are called spirilia. Some of these, again, can aggregate together and form those stalk structures or the filaments, but most of them exist as single cells. They have a plasma membrane that is encased within a cell wall, so similar to our plants, they have a membrane with a cell wall. That cell wall, though, is different than in plants. It's not made up of cellulose. Instead, it's made up of a compound, a carbohydrate called peptidoglycan. And this is a common target of antibiotics that prevent them because we don't make peptidoglycan. So destroying it or preventing it from working impacts bacteria, but not us. They're divided into two classes, gram-positive or gram-negative, based on a staining technique for identifying them. So gram-negative bacteria, they have a thinner cell wall, so thinner peptidoglycan. And then on the outside of that, in addition to their plasma membrane, they have another outer membrane made up of those phospholipids. And that prevents the cell wall from taking up what this gram stain. So it causes it to be a specific color, whereas the gram-positives can be another color. So that's how we differentiate them. But it's due to that smaller cell wall and that outer membrane. That also, though, makes them more resistant to antibiotics, especially things like penicillin. Whereas the gram-positive bacteria, they don't have that extra outer membrane, and their cell wall is much thicker. So they have a lot more peptidoglycan and no extra layer on the outside. So they're more sensitive to antibiotics, and they take up the stain during the staining procedure. So they are typically purple, and then gram-negatives are pink. So they're easily distinguished by doing this simple staining technique. Some of the other features that we can see, many of them have a capsule, which is a sugary layer on the outside. It helps them attach. Some of them also have a flagella that we'd seen on the previous slide that helps them to move or swim. Some have shorter projections called pili that help them attach. They also can exchange genetic information in some types. And some of them create a really dormant state called an endospore. So this encloses their DNA and just a little bit of cytoplasm, but they are extremely resistant to environmental stresses. So like to boiling, to high temperatures, to sunlight or UV. Again, they're very, very resistant, so they can exist for a long time. And this is the problem and why you need to get a tetanus shot. So tetanus is caused by an organism that can form these endospores that lives, then they hang out in soil. So if you have a cut and you get some dirt and some of these endospores in the cut, then they're in a nice healthy environment. They start reproducing and cause an infection. So again, these endospores are a dormant state. They're not reproducing, but they can cause infection. Now, the way that these organisms reproduce is a process called binary fission. So they replicate their DNA and then essentially divide the cell into two so that each cell gets gets its own DNA message. They can also go through this cool process called conjugation, where they can exchange genetic information. It's similar to like bacteria sex that's happening. So that pilus, one of those short projections, attaches to two different bacteria cells, and the DNA actually moves between that pilus from one cell to another through an area called the conjunction bridge. Where this is significant, it's not a way to reproduce, but exchanging this information is one of the issues we contend with antibiotic resistance. So it's one way that one bacteria that could be resistant to an antibiotic can share that resistance with another cell, is through this process of conjugation. So our bacterial conjugation shown here, again, here's this conjugation bridge, and what's happening is we see this piece of DNA that is moving from one cell to the other. So by the end of conjugation, both of these cells now have this extra DNA, typically referred to as a plasmid. And if there's an antibiotic resistance gene that makes a protein that makes them resistant to an antibiotic, now both of these cells have that resistance. Some of the other features, typically they are small, again, simply organized, no organelles. Again, they lack a nucleus. They are different from eukaryotes because, again, no internal compartmentalization, no organelles. They're much smaller than eukaryotes. They're typically single-celled. Eukaryotes are usually multi-celled, but not all of them. They have many fewer chromosomes. They use that binary fission, just a very simple copy the DNA, divide the DNA. Eukaryotes go through mitosis or meiosis, the more complex process. Many prokaryotes have a flagella that most eukaryotes do not, and some metabolic diversity. So they go through some different types of ways to get and use energy. Again, just a nice little comparison thing here. Again, our prokaryotic cells, again, much smaller compared to the eukaryotic. Again, lots of organelles inside of our eukaryotic, not seen in the prokaryotic. Again, unicellular compared to having multiple cells. One prokaryotic chromosome, one piece of DNA, whereas we have multiple chromosomes that are linear, and we have for us, we have 46 chromosomes compared to one in this prokaryotic cell. So much less DNA contained inside of it. Again, that cell division with binary fission here, again, the cell simply just divides into two. Whereas in mitosis, the cell, all of the chromosomes have to be divided through this process of mitosis. Again, the presence of a flagella is common in bacteria, not so much in eukaryotes. And then their diversity. So we'll talk about some different ways they can get energy, like chemoautotrophs. So leading into that, there are four main methods that drive how cells get and use carbon and energy. And that sets up all of these crazy terms. So first of all, the difference between an autotroph and a heterotroph. So auto or self, this means they can obtain their own carbon from an inorganic carbon, which is carbon dioxide. Whereas heterotrophs, they need organic molecules. They cannot get carbon from carbon dioxide. They need an organic molecule that has carbon and hydrogen in it, like an amino acid or glucose or something to that effect. So they can't use carbon dioxide for carbon. Now the other differentiation is where their energy comes from. So those that use light energy have photo added on, so that's sunlight energy. Those that use chemicals are called chemo organisms, so they are using some type of molecule to get energy using electrons and oxidation reduction. So we get these four categories whenever we put all these possibilities together. All right, so photoautotrophs, so the photo equals sunlight. They're using sunlight for energy. And then autotroph tells us that they're using carbon dioxide. So these organisms use sunlight for energy, carbon dioxide for carbon. Chemoautotrophs use chemicals like inorganic chemicals that they oxidize or get electrons from. That gives them energy. And they also are getting their carbon from carbon dioxide. Now we also have photoheterotrophs. So photo, again, they're getting their energy from sun, but they're getting their carbon from organic molecules. And then chemoheterotrophs, again, they're using organic molecules for everything. So they're getting their energy from organic molecules and they are getting their carbon from organic molecules. And this is where we fall. We need food and organic molecules to provide carbon to make other proteins and other amino acids, other carbohydrates, and we need organic molecules to give us energy. So going through respiration to break down the glucose to release that energy. Now, prokaryotic cells are very important for us. They play many roles. Many of them are decomposers, so they help break down nitrates and various organisms and compounds in our cells, dead material. They fix nitrogen, meaning they can take nitrogen from like ammonia and nitrogen gas, and they can use it to make compounds we can actually use, like amino acids. Commercially, they're genetically modified to do lots of things. They can be modified to make things like insulin for diabetic therapy. They can be modified to actually break down different oils in a process called bioremediation. But of course, many of them or some of them can cause disease, so they have a huge importance to our health, unfortunately in this case in a negative way. And this is just showing that bioremediation. Here they're actually spraying bacteria into this polluted area, and the bacteria actually break down these toxins in this oil to restore that area back to its original habitat. Now, just to talk a little bit about the other type of prokaryotic organisms, the archaea. These are, or archaea, the main ones we'll talk about, methanogens and extremophiles. Methanogens, if you're familiar with climate change, one of the greenhouse gases that allow us to live on Earth, that trap the sunlight energy to warm our atmosphere. But too much of those greenhouse gases is causing the temperature to increase, which is leading to climate change. The main contributor to that is carbon dioxide, but another one is methane. So these methanogens are these prokaryotic organisms. They use hydrogen gas, and they reduce that hydrogen gas to reduce carbon dioxide into methane. And then that methane gets released into the atmosphere, and that again can lead to that increase in temperature because too much methane traps too much heat. So they are anaerobic. They don't survive in oxygen, but where these are found are in ruminants. So like our cows and sheep, in the stomachs where it's anaerobic, there's no oxygen there, but they're using the food that's available in the gut of these organisms, but they're releasing methane as a byproduct. So this is the main contributor to the increased amount of methane in our atmosphere, is from these methanogens. And then just some others that are pretty cool. Extremophiles live in very harsh environments where we wouldn't think anything survives. For example, thermoacidophiles, so they love high temperatures and they love acid environments like sulfur springs. Organisms that live like at the depth of the ocean where it's very cold, there's no sun, there's no oxygen. Again, this is where we would expect to find some of these extremophiles. And this is an example of one of the hot sulfur springs where these thermoacidophiles would actually be thriving in this condition. Now back to our bacteria. Most of these bacteria vary in their lifestyles. So for example, cyanobacteria, they're thought to be the first organisms to go through photosynthesis. Again, they're one of the filamentous, meaning they have multiple cells. They're multicellular, even though most bacteria are single-celled. But they fix nitrogen. So they take nitrogen from the atmosphere as far as nitrogen gas. So they make it to usable forms that we can use. And this and other organisms using this are called heterocysts. And many of these bacteria are also decomposers. Some of them form, though, what are called biofilms, which is kind of a community. They secrete a carbohydrate, a sugar, and other bacteria and other organisms stick to them. That film on your teeth in the morning, that's an example of a biofilm. But they are very hard to treat. They're resistant to antibiotics because they're kind of hidden. So even if you get the outer microbes, the inner ones are still going. So this can be problematic in treating disease as well. And then just another one here, tuberculosis and other, again, diseases that affect organisms, of course, come from these prokaryotic bacteria. Here's just an example of that cyanobacteria. This is Anabaena. Again, just multiple coccus-shaped cells that form these filaments to make up this organism. Here's a list of various bacterial diseases. You are not going to have to know all of these. Again, just kind of bringing some of the more common ones. So like anthrax, this is one that forms those spores. There was actually used in bioterrorism where the spores were put on an envelope and sent to somebody in government as a way of trying to cause an infection. Botulism that's in some canned food. There are some STDs like chlamydia, cholera that had a huge impact in contaminated water, cavities, diphtheria, gonorrhea, another STD, leprosy, Lyme's disease is becoming more prevalent, spread by ticks, especially from deer to humans. Ulcers are actually caused by a bacteria called Helicobacter pylori. The plague, pneumonia, tuberculosis, typhoid fever, and typhus. These have all been ones that have hugely impacted, caused a lot of death or a lot of disease over the years. Of course, many, many others out there, but just kind of highlighting a few more common ones. All right, now switching into viruses. Now, viruses are not considered living. They hugely impact our life. They are not prokaryotic, though, because they are not cells. Therefore, they are not living, but they do hugely impact our life. So viruses, again, parasitic chemicals. Again, they're not cells, they're not organisms. So they're parasitic chemicals. They have pieces of genetic material, either DNA or RNA, could be either, wrapped in a protein called a capsid. Again, they are not alive because they do not possess all of the characteristics of being alive that an organism has. They don't have a cell. They can't reproduce on their own. They're extremely small, much smaller than prokaryotic cells, and they can infect all different types of organisms. So even bacteria get infected by viruses. Plants get infected by viruses. Fungi get infected by viruses. It's not just humans. Now, some of them on the outside of their capsid may have a membrane like our plasma membrane called an envelope that has proteins and lipids on it. But there are many different structural differences between different types of viruses, so a lot of variability here. Just to show a few examples, excuse me, of what these can look like. Bacteriophages, which are actually viruses that infect bacteria. They have this weird kind of alien shape to them. Here's an example of TMV, tobacco mosaic virus. It infects tobacco plants. And then one for humans, HIV. So again, huge variety of what these viruses can actually look like. What they have in common, though, is they have a genetic material, DNA or RNA, surrounded by some type of protein. And some of them, like the one with HIV, also has this envelope on the outside, but not all of them. So a little bit about these. So again, bacteriophages, these are viruses that infect bacteria. Again, they're structurally and functionally very diverse. They don't all look the same, but just kind of looking at an example of how they infect bacteria. A T4 bacteriophage, its tail fibers actually attach to the surface of a bacteria and they pierce the cell wall. So they kind of poke a hole in it. And then mostly the DNA gets injected into the cytoplasm. So the whole thing isn't going in, that whole little alien thing doesn't go inside of the bacteria. just the virus's DNA. Then that virus DNA gets transcribed and translated using the bacterial cell. So it uses the enzymes the bacteria already has because it goes through transcription and translation, but now it's making viral proteins. And then those components get assembled and it causes that bacterial cell to rupture and it releases the new viral cells that then can affect other bacterial cells. So again, just kind of looking at this. So again, these tail fibers that stick down here, these little spider-like projections, they attach to the outside surface of the bacteria, and then they only inject the viral DNA. Again, the whole thing isn't going in, just that DNA enters, but then it can go through transcription and translation. But now that cell is making viral proteins, not bacterial proteins. And then it ruptures the cell and it releases new bacteriophages. Now, how they can enter prokaryotic cells, because some of them, of course, can, like these, can enter bacteria. So during the viral infection, the virus cycles either called lytic or lysogenic. A lytic cycle, this is whenever the virus kills the infected cell that it's replicating in. So it lyses the cell, makes it pop open. So of course that cell is not going to survive. It will die. However, it can go through what's called a lysogenic cycle instead, where instead of the DNA kind of taking over, it actually gets integrated into the nucleic acid of the host cell. So now the host cell has its own DNA, but it has some extra DNA from the virus, and that process is called lysogeny. And now the new DNA is called a prophage because it has some viral DNA added in to the host genome. And this is also called gene conversion, which is the expression of those viral genes along with the host genome. So it doesn't completely take over. There's more of a marriage of their DNA. And that process is being shown here. So again, this is the lysis or the lytic cycle. So that bacteriophage comes in, it releases its DNA, it starts replicating. Those new bacteriophage ultimately get assembled, and then the cell gets lysed or popped open, and new bacteriophages go out. Now, sometimes though, that DNA, instead of taking over and lysing the cell, sometimes that DNA actually gets added into the host genome. And this is that lysogenic cycle, where they just kind of hang out together. They live in harmony here for a little bit, but then it can go through a process where it starts reproducing and replicating and could still ultimately cause lysis of that cell. Now, in animal viruses, it's a little bit different. So animal viruses enter the cell by fusing to the membrane. So again, that envelope that some of them have, that membrane just fuses with our membrane, or it induces endocytosis. So kind of like phagocytosis, where the cell engulfs food or something. Here it's engulfing the virus. So on the surface of the animal virus, instead of those tail fibers, there are proteins called spikes. If they match, again, that's a big if, but if they match the animal surface, like a receptor or something, it triggers that membrane fusion. And then once that's inside the animal cell, the virus sheds that coat, that capsid, and it releases its DNA or RNA. And then just like with the bacteriophages, it hijacks the cell, and the cell now starts making viruses, and it typically exits by bursting and killing the animal cell. So this process here, again, HIV is happening. So again, these little spikes on the outside of the cell, they attach to a receptor or something on the animal cell membrane, and it causes fusion. So again, it's just essentially going to be taken in, releasing that capsid, and then ultimately releasing the viral DNA or RNA. In the case of HIV, it's an RNA virus. So then what's unique about HIV is it's RNA, so it has to be converted to DNA before our cells know what to do with it. So it has its own enzyme called reverse transcriptase. We don't make this because we do DNA, RNA, protein. But in this case, they're using RNA to make DNA that then is transcribed and translated. So they need this extra enzyme called reverse transcriptase, and this is one of the main enzymes that's targeted in HIV treatment. But once it's able to do that, once the viral DNA is made, then it's transcribed and translated at those ribosomes to make viral proteins. They all get packaged, and then either it lyses from the cell like we see down here, or just like it came in, instead of going through endocytosis, it can go through exocytosis. So here the cell remains intact. This would not kill the cell, but it releases that enveloped virus without killing the cell. Others, though, cause lysis that rupture the cell and leave it, of course, dead after infection. So some of the diseases caused by viruses, more of the emerging ones. So influenza, we hear a lot about the flu. This is one that's had the most lethal, has been one of the most lethal viruses to human history by far. So there have been many different strains of influenza. There still continue to be different strains of influenza like bird flu and swine flu, and we just keep seeing kind of mutations and reemergences of it. AIDS and HIV, again, the one that we just went over there, ultimately came from chimpanzees and monkeys. Ebola is one that started in Africa that, though, has now can attack human connective tissue. The Zika virus, transmitted by those mosquitoes. The SARS virus, which is one of the respiratory, and that's where COVID is actually a relative to the SARS. Of course, it's greatly impacted health. So again, this is not anywhere close to an extensive list, but just some of the newer viruses that are impacting our health. Some other important ones, of course, chickenpox, now that we have a vaccine against, a little less, and it's not a fatal one. Ebola, again, we already mentioned. Hepatitis is another, especially if you work in the healthcare system, you get vaccinated against different hepatitis species. So there's hepatitis A, B, and C. Herpes, some of our STDs, again, influenza, one of the most ones that have impacted or caused death in our history. But again, lots of different ones. Now these organisms don't always cross from one animal to another. So they have to be able to bind to the cell in order to get in. So that's what's going to limit or determine what cell or what organism they can infect. They have to first attach. So if they don't attach to our cells, if our cells don't recognize them, then they can't cause an infection. Just a few others here: measles, polio, which has pretty well been eradicated. Rabies is one that can cross a lot of species, so between a lot of different animals to humans, whereas others do not. Again, they're only found in humans, or they're only found in one specific animal. And again, that's due to their ability to attach to our cells. All right, and just again, a few other: smallpox, of course, the SARS, the coronavirus like COVID, yellow fever is another. So many of these have been eradicated, or we have vaccines against, but then unfortunately still many newer ones emerging as these viruses can mutate.